that's the annoying part.
Yeah, there's a lot of steps to go through.
All right, thank you so much.
No problem. Sorry it took a while to take it over here, but hopefully it doesn't delay things too much.
I give them the quiz, so.
Thank you.
Thank you very much. 啊，谢谢啊。I haven't prepared this. One second. Okay. All right, finally we get this out. Everybody's happy with the quiz? I plan to do that at the end of the class and give you a surprise. So the surprise comes a little earlier. Okay, so I hope everybody enjoyed the tour last week. And yeah, it's a good machine, and you have seen the lab operation. I think this is important, especially for the biology and for the computational students. And you probably haven't had a lot of chance to see the lab, and that is what the environment is. Okay, so we'll start from today is talking about the DNA sequencing part. And so before we get into that, let me briefly overview what we talked about 2 weeks ago. So the first point I want to make is for the next generation sequencing analysis, there are actually 3 different steps. The first one we call the primary analysis. That is basically from those imaging signals or from the electrical changes and to turn into the ACGTs. We usually don't take care of that part. The sequencer comes with the algorithm to do that. The secondary analysis starts from the data QC, and then we need to do the sequence alignment or do the assembly. It depends on different experiment. But the secondary analysis part, you do not need to know too much about the experimental design. So meaning that as long as you know this is RNA sequencing, you have specific pipelines to do the alignment, that will be fine. You do not need to know the first sample is control, the second sample is treatment 1, and things like that. Okay, and in the last lecture, we spent some time talking about sequencing alignment and those type of stuff, and those are all fall into the secondary analysis. And the third tertiary analysis is the part you really need to know the experimental design. You need to know what are all the biological questions you want to achieve. And then by combining all these things together, and you will be able to get the analysis done. So for the tertiary analysis, each individual type of applications, you have a specific set of tools that we need to use. And this part need to have the computer person, the bioinformatician, potentially statistician, and more importantly, the biologist in team to understand the results. And we also talk about the concept of the quality. And actually for the sequencing, there are 3 levels of quality. The first one is the base quality. So every time that the sequencer reported ACGT, and what is my confidence level of that measurement is correct. If I report it is A, is that really A? What's the chance that I make a mistake? So those are called the base quality. And for every nucleotide, this sequencer's output, you will have a base quality that is associated to that. The second level is the mapping quality. So when I have one sequencing race, I map that back to the reference genome. What are the chances I made a mistake? Okay, and then that will be the mapping quality. So we will have one for every single sequencing race, and this is usually reported by the sequencing alignment algorithms. And the third level of quality is consensus quality so this is the part is when we call there is a genetic variance there. So for example, this part I say there is seems to have a homozygous variance, and this is a heterozygous variance. What are the chances I'm making mistake? So this is the third level of quality. So at the time that you discuss with your collaborators about the data quality, and you need to be very clear which level you're talking about. And we started to talk about different type of alignment algorithm that we can use to do the next generation sequencing. We didn't really specific talking about dynamic programming, but this is really everything that for the alignment algorithm series started. And there's two concepts. One is the global alignment, the other one is local alignment. From the figure, you can see global alignment, you are trying to align two strings of sequence from the beginning to the end, while the local alignment, you want to really only looking for a very specific part of it. And the algorithm to do the global alignment is Needleman-Wunsch, and for the local alignment is Smith-Waterman. We didn't specifically talk about that. If you're interested in, and I think if you're an informatics student, you should already know this. And if you're a biologist, then you can go to look for some videos. I'm sure there are a lot of things on YouTube about this. And then we start to talk about different categories of alignment algorithms. So there is a hash table-based alignment algorithm, like the seed extension-based approach. Remember, we have a few slides talking about that. But we spend most of our time talking about the Burrow-Wheeler transformation. And just a little bit summary of that. I'm not going to go through this with you again. But the general point is the Burrow-Wheeler transformation. The first one is this is a reversible conversion. So it's unlike a sort. If you do sort, you cannot do unsort because everything is going to be lost. But for the Burrow-Wheeler transformation, you do the conversion and you can convert it back. I think one of the quiz questions is to convert it, the other one is to convert it back. And another very interesting feature of this is the index size of the Burrow-Wheeler transformation is very small. In a sense, it's the same as your reference genome. So for human, we have 3 billion letters, and in theory, you just have 3 billion letters about the index size. That is very, very small. And the second part is a very, very useful feature is the certain complexity. It is proportional to the size of the query sequence. So if you have a 75 base rate, and that's the amount of computational time you need. It's not n squared, it's not NP hard, and it's very, very fast. You cannot get in faster than that. Okay, so many algorithms are using this Burrow-Wheeler transformation, and the most important ones will be the bow tie and the BW. It should be BWA. I made the typo there. But again, these are how this will work, and I'm not going to go through this again. And if you have trouble with it, and go back to the YouTube video. By the way, everybody have access to the YouTube video, right? I try to put it there. So you should be able to figure it out. And the last point that I talked about in the last lecture was the standard data formats. For the next generation sequencing, you can make your own file formats, whatever you do, it doesn't really matter. But the standard formats are FASTQ files, the BAM or SAM files, and the VCF files. This is really corresponding to what we talk about the 3 levels of quality. And if you think of the FASTQ files, these are just the ACGTs and the base quality for every single one of it. And the SAM and BAM, this is the information tell you for each individual race how the alignment goes and what are the alignment quality, and most of the information that related how well this particular sequencing race aligned to the reference genome. And the VCF file is about individual locations, their genetic variants being identified. Okay, so I hope that if you are still not familiar with this concept, go back to your lecture notes or get a few of those samples to take a look at. And this is going to be the bread and butter of the data that you're going to navigate through. And we actually talk about visualization tools a little bit. So basically, there are two things that at least my group are using very regularly. One is the UCSC Genome Browser, and this tool, the nice feature of this tool is not only they provide a huge amount of annotations. So basically, every single genomic location and what are the genetic function that we know of, and whether there's a SNP there, what are the conservation levels, is this in the introns or exons of the gene? Every information that is available is supposed to have a track on that. So you can visualize that what individual base that is about our knowledge. And more importantly, that we can load our own next generation sequencing data into it without too much cost. And actually, this tool is very interesting to see it's evolving and in a very early stage of NGS world, and we will have to load our sequencing data back into the UCSC Genome Browser, and they will host that for a couple of days, and you can visualize that. And later, they figure out a way to do this in a remote fashion. Now it's becoming very, very convenient. And the second tool is called the Integrated Genomic Viewer, and developed by the Broad Institute. And it's very, very similar, and it's Java-based. You can install on your own desktop, and then it provides a lot of very unique features as well. So sometimes you can do heat map using this, and they also have the module to look at the splicing outcome of your own data. So just to play with that is very interesting. Okay, so that's what we talked about 2 weeks ago, and remember that was an incredibly intense 180 minutes. And but in the next 3 lectures from today, we are going to focus our discussion on the DNA sequencing part. So today, I'm going to most focus on the biological components of it and giving you a few examples, applications. In most of cases, it's not going to be as heavy as last time. It's just for your information, which is good. But for the next one, and when we go through the bioinformatics and statistical components, it becomes really, really heavy. There are mathematical formulas and there are different algorithms, so just be prepared for this part. And then on the September the 29th, and we were talking about once you identify the genetic variants, how are we going to interpret it? Meaning that, so what are their functions? What are their predicted functions? So we'll spend a full lecture to discuss that part. And then we will have a guest lecture coming in, Dr. Matteo Vata, and he is an adjunct faculty in our department of medical genetics, and he does a lot of clinical work. So a couple of years ago, actually, he set up the clinical sequencing laboratory at IU Medical School. And so he will come here to give a very detailed discussion on using this technology in the clinical setting and what are the requirements, what are the differences with what we are doing in the research settings. So it's going to be a very interesting topic. Okay, so what I'm going to discuss today is the first part is give you a little bit of definition on the genetic variants that what we are talking about. And then review a little bit on the state of genetics of complex disease before we have the next generation sequencing technology. It has been a field for many, many years, and we only have the sequencing technology for less than 10 years. And we are talking about the next generation sequencing and some of the examples. And I will give one example of using the sequencing technology to really try to understand the etiology of the disease. And now there are many, many of such examples, right? So if you open the journal the Nature Genetics, every issue they will have a few of those articles coming out using the technology to understand disease. But I'm going to give you one example, and also that talking about a little bit on the implications on the personal genome sequencing. And towards the end, I'm going to talk about the experimental considerations of DNA sequencing with different scopes. Okay, so the first topic is about the genetic variants. Just a little bit of clarification of the concept of what we are trying to identify here. So there are different types of genetic variants, actually. The first part is called SNP. It's a single nucleotide polymorphism. And it's very, very simple. It's just one nucleotide difference of your genome comparing to the reference genome, or the difference between two people. So there's nothing fancy about it. So the second one is actually there's a short insertion deletions, or we call it indels. And it's very simple. It looks like this. So you see that there is in the reference genome, there's missing 3 letters. But in your genome, there's 3 more letters. So this is not a single nucleotide change, rather than there is a few more or a few less of nucleotides in the middle of the genome. So this is called the small insertion deletions. Actually, in our genome, about 82% of the genetic variants are the SNPs, and the remaining 18%, and almost exclusively, they are the small insertion deletions. There are larger ones, but the number will be very, very small. And then there's if we go a little bit, increase the magnitude a little bit, there is a microsatellites. So you see the definition of that is 2 to 6 base pair repeat units like this TCA, 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 TCA for many, many nucleotides. So this is called a microsatellites. And then further, there will be something called mini satellites, which means those repeat units are become bigger, 10 to 100 bases, but they occur again and again. So those are called mini satellites. And then there are some others that there's a repeat genes. So the entire genes have multiple copies in the genome. So you can see the scale become larger and larger. And of course, there are large structure variations, including insertion deletions. For this part, we are talking about long insertion deletions. In the next lecture, we'll talk about the informatics considerations to identify those long insertion deletions as well. And there are duplications, there are inversions, there are copy number variations. There are many, many different types of larger structure variations. Coming back to the SNPs, a few more words about that. So the SNP is defined as a single base change in the DNA sequence that occurs in a significant proportion of a large population. Okay, so if you read this word, before you read this, you probably think, oh, I know what the SNP is. It is a single nucleotide changes. Actually, it's wrong technically. So in order to qualify that as a P, which is a polymorphism, so you can see you need to have a significant proportion of a large population that carry that variance, and that is called a SNP. So meaning that if that is a SNP or a single nucleotide change that only you have it, it's called a single nucleotide variance. Okay, you cannot call it polymorphism. Okay, in the past, you need to at least have 1% of a population has it to define that as a polymorphism. Now people start to, relax that requirement a little bit. If you have, as long as it's a significant proportion of population, that will be fine. So in general, when I write papers and when I write grants, I usually do not write SNPs anymore. I just write SNE, which stands for the single nucleotide variance. Okay, and some statistics about the SNPs, and it will occur in 1 in every 300 base of the human genome. So in our genome, there are more than 10 million SNPs across our human genome. And SNPs are conserved across the genome often in a pattern called haplotypes. So what that means is if you have a SNP here and you have a SNP here, and they are not independent to each other. So because of our evolution, the genes that kind of being altered during the evolution process, but some of the SNPs, if they are close to each other, they often coming together. So, for example, the first location is A or C, the second is G or A, and these two SNPs, they are not independent. A may always paired with G on the other location. In a sense, if you know one of it, you already know the other one. So that is called the haplotype, and the region that defines that is called haplotype block. We'll come back to this concept in the later slides as well. And again, I told you the statistics about 82% of the genetic variants in human genome are SNPs. And there's another very good, important concept about SNPs, and a lot of people probably didn't pay attention to, and this will be a very good exam question to ask as well. So that's a hint, okay? So SNPs, there are two types. One is called transitions. The other one is called transversions. So what transition is a substitution between the purine or between the chromatins. Okay, so meaning that A and G, those changes are called transition, and the C and the T, those changes are called transition. While transversion is the substitutions between a purine and a chromatin. So A to C or T is called a transversion, and the G to C to T, and so on and so forth. Okay, so these are a very, very important concept, a definition about these SNPs, transition, transversion. And actually, it's very useful for us to look at the quality of our data. So look at this statement. The ratio of the number of transition to the number of transversion for a pair of sequences, it should be, let's say, all the mutations are random. You can become anything, okay? And our ratio of transition and the transversion should be 0.5, right? So why is that? Because for transition, for one nucleotide, you only have one option to change to. Well, for the transversion, you have two options to change it to. So if everything is random, this ratio, or called transition transversion ratio, should be 0.5. Okay? But in reality, when we look at the human genome, across the entire genome, the transition transversion ratio is usually 2 to 2.1. And in the exon region, is 2.8 to 3.0. Okay, actually, we have much more transition comparing to transversions. And the reason for that is really for the evolution issues, that some of the changes are more dramatic than others, and those individuals or species are not going to move forward. So it will be eliminated by the evolution. But this fact is not 0.5, rather it's 2 to 2.1 or 2.8 to 3.0. It's something that we look at for every of our whole exome sequencing data or whole genome sequencing data. So we will calculate this transition transversion ratio after we complete the experiment. So if you tell me that after your experiment, you found the transition transversion ratio is 0.5, I would say there are a lot of things go wrong. I don't know what goes wrong, but there are a lot of things goes wrong. It's not possible. And then this is going to be a very important metric I often look at after we got the data. That makes sense? All right, just pay attention to this fact, and then I will guarantee we'll have this question in the exam. And then, so that is the concept about SNPs, and some of them you probably already know, others you haven't paid attention to in the past. So now let's go to the another type of variation, which is the structure variation. It's traditionally defined as deletions, insertions, or inversions that is larger than 1 kb. So it's not going to be just one nucleotide change or a few dozen nucleotide change. It has to be really large, like a few thousand bases or sometimes even beyond. It often involves the repetitive regions of the genome and complex rearrangement, and there's a lot of such things in cancer as well. And then usually in the past, before we have the sequencing technology, there's really no optimal method for identifying these structure variations. But because we have the sequencing technology, and now we at least have the information, have a hope to identify those. And again, next lecture, we'll spend a few slides talking about the algorithm to identify these structure variations. And it affects a larger fraction of the genome as compared with SNPs. And so that is functionally, it's probably have a more impact on the phenotypes. But another interesting fact is the analysis of 1,000 genomes project pilot data identified over 1,000 structure variations that can affect the coding sequence. Why I want to point this out? It's because 1,000 genomes, individuals, they do not have a clear disease phenotype. They are healthy individuals, at least at the time they were being sequenced. So which means those structure variations are very, very important, but it's not guaranteed that you detect one and it will kill that individual. No. So in our normal populations, we also have a lot of structure variations, and there's many biology we need to understand about that. And there's a few things that I want to mention before we go into the details of each individual technology. So the first one is most genetic variants, actually, they do not have effects, okay? And so that's what I said, right? They're called silent variations. They do not have a phenotypical effect. And many of them, they do not occur in the coding or regulatory regions, so they don't really change the gene expression levels or gene regulation process or the protein functions. So they are just in the remote desert part of the genome. Or even within these regions, it's very likely they do not have any effects. And we will revisit this whole concept 2 lectures from today. So when we talk about the interpretation of the genetic variants being identified. Although they do not have phenotypical effects, and we cannot say they are not useful. And in many cases, they could be useful for the forensic science and for the paternity test, archaeology, and ethnic migrations. And sometimes, more importantly, they can be used as a criterion to select the individual patient from certain clinical trials. So they can be very, very important, although functionally they may not have a phenotypical impact. So I think in the first lecture, we talk about the forensic science and things like that. So those are very, very important as well. So since most of the genetic variants are SNPs, so there are a few concepts about the SNPs. Again, we will talk about this in much more detail 2 lectures from today. But in terms of SNPs, there are many different types. So you can see there is something called a nonsense SNP. Nonsense is, you often hear about these terms, so we need to make sure that we really understand what that means. It's very simple. It's within an exon and being translated amino acid change to stop codon. That is called nonsense. So basically, when the translation goes, and then suddenly it's supposed to be a normal amino acid, and then the translation should go on. But because of the one variant, they change that into a stop codon. If that happened, you cannot further do the translation, and you will have a much more truncated protein that is either resulting in altered functionality or triggers other pathways like non-sense-mediated decay to kill the gene. And frame shift is more referring to the insertion deletions. It's within the exon and translated insertion or deletions interrupts the reading frames. Okay, we'll get back into that a little bit. Actually, my group have several papers about this frame shift and then insertion deletions. And next one is the coding but non-synonymous SNPs. So what that is it's very similar to nonsense, but instead of changing it to a stop codon, you change that to a different amino acid. So that will also potentially change the protein functions. There is a splice 5 or splice 3, which is related to the splicing part, and in the first 2 base or the last 2 base of intron, and we know those regions are very important for splicing recognition. And the coding and is synonymous, that is in the coding region, but does not change the amino acid sequence. So in many cases, a lot of people consider they are not very functionally important. We'll also challenge that notion, but we'll talk about it in the later lectures. UTR regions, either in the 5' UTR or 3' UTRs. We know that in the 5' UTR region, they are very enriched in the translational control signals. And then in the 3' UTR regions, those are hotspots for the microRNA binding sites. So all this, if there's a variance in that can impact the translation or degradation of the gene or protein. And then the near gene 5 or near gene 3 prime regions, those are usually the promoter regions or enhancer regions that control the gene regulation, and there are also the SNPs in the intronic regions. So at one point, that after we identify those SNPs, we need to subcategorize those, what are different functions of different SNPs. Okay? Any questions so far? We will only have one break today, so because we had a quiz. So now let me go through a few things regarding the state of genetics of complex disease before we have the high-throughput sequencing technology. And this used to be a historical thing, and then people stop updating it. But you can just for your information, I'm going to use this panel here. So you can see that this is the year from 1870 to 2009. Actually, it's already a few years ago. But you can see that history in 1950s, Watson and Crick discovered the double helix of the DNA. And then here is the sequencing capacity. In 1950s, each year, one person, your ability to decipher the genetic variant sequence is about 1 nucleotide a year, okay, one person. And then fast forward to 1970s, and that is where the Sanger sequencing, the other type of sequencings were being invented. So you can see that one person in a year can get to about 1,000 to 10,000 bases. And then we are talking about 1990, that is the beginning stage of the Human Genome Project. It's about 200,000 nucleotides per year per person. And then in 2009, and there's more zeros than I can count. And today there's a few more zeros afterwards. But the point is our sequencing capacity certainly increased dramatically. And we talked about this before, and it's very expensive, and it costs you $8,000 to sequence 1 million base in 2001. But today, it only costs about 2 cents in my lab. Some places probably even cheaper. Okay, so those are the technology part. But in terms of understanding the genetics, before we had the sequencing technology, people have studied genetics forever, right, for many, many years. And initially, what they are relying on, there is only the microsatellite markers. Remember, we talked about microsatellite in the earlier slides, 2 to 6 nucleotide, and then repeat many times. That the number of repeats in the microsatellites serves as an important genetic marker, and people is trying to use that to do either family-based study or association studies. And then later, the SNP array being invented, and you can see that earlier stage is about 6,000 SNPs across the whole genome. That is pretty good. And then eventually become 100,000, and then become 1 million. I think today the highest density one is about 5 million SNP markers on the array, and the normally used one is about 1 to 2 million SNP markers on the array. And these are, again, not a sequencing-based technology at all. Those are microarray-based in many cases. And when people do the analysis in the early stage, and because of the SNP marker is not that dense, and people often do the linkage analysis, and then later we'll all move to association studies. I think many of you heard about the GWAS, which is the Genome-wide association studies. We'll get into a little bit, but those are the analysis method before we have the next generation sequencing technology. So now I'm going to spend a few slides talking about the association study part, and basically trying to set up the stage to see that what are the additional information we can get after beyond the GWAS using the sequencing-based technology. So the GWAS and also stands for Genome-wide association studies, and they are commonly used to link genetic variations with disease traits. So these are, the study design can be really complicated, but the general idea is it's basically a statistical game, right? So you got two group of individuals, and this is the case. Usually that represents people who are sick, who have the disease, and there is a control. I'm going to use this panel. That one's too far away from me. So you can see this is the control, okay? And then for certain disease, for certain SNP, so for the first one here, the SNP, and we are seeing that if this is a purple, that means that individual doesn't carry the SNP. And then, or this is a yellowish, and that will mean it's heterozygous variants or homozygous variants with different colors. But from this, you can pretty much see this group is much more colorful than this group. That means that there is this particular SNP, the occurrence of this SNP is associating with the disease you are studying. So it's very, very simple statistical game. And the second SNP, so you can see very similar patterns. Okay, so the association study is really, there are a lot of things that we need to consider, but the general idea is to see how the SNP is going to be associated to the phenotype. If genetic variants are more frequent in persons with the disease, the variation are said to be associated with that disease. Okay, so I'm sure that many of you that have seen this figure multiple times, and this was a very nice review paper a couple of years ago, published in Nature, basically trying to see that how the NGS will take us forward comparing to the GWAS studies. So the genome-wide association study, if we do that using this association-based study, okay, this is important. Is based on the assumption is a common disease is caused by the common variants. It's a common disease, common variants. I shouldn't say cause, but common disease, common variants assumption. So what that means is if you look at this particular figure, right? So X here corresponding to the allele frequency. So what that means is if we look at one variance, in that spectrum, in the right side of this panel, and those means that variance, it's very common in the general population. A lot of people has it, and 5%, 10%, 20% people have that genetic variance. And the Y here is called effect size. So what that means is if I have this variance, and my probability my risk to get into this disease is like 10 times higher than the person who doesn't have this variance. So that is the effect size. Or we can say that if I have this variance, my risk to get to this disease is 1.1 times higher than the people who doesn't. And then that effect size will be very, very small, right? So what we're trying to look at is some of the variants, they're going to be, effect size is going to be very, very large, others are going to be small. But if you look at this whole spectrum, if we see this right top corner here, and those are the ones that not only very, very common in our general population, but also the effect size is very large. Those variants usually will be associated to the phenotype that is not life-threatening, like our eye colors, hair colors, and this type of stuff. And usually we already know those genes, okay? And then, I guess, I'm going to use this. We got a lot of technical challenges today. And for the GWAS, usually we're focusing on this panel, this part, the Genome-wide association study. And those are the ones that it's very common in the general population. However, the effect size is much smaller, okay? And also that there are some of other variants in this spectrum. Those are very rare variants. You can see the effect size, the frequency in general population is it's very rare. But if you have it, you have a large proportional possibility to get to this disease. So these are very, very important as well. So in the past, this part we have to do linkage type of family-based study in order to collect enough individuals that carry the genetic marker. And for the GWAS, it will be focused on this panel. But using the sequencing-based technology, we can go through the entire spectrum of this. So that is the general idea. But one thing I want you to really remember in this slides is GWAS, the assumption, the underlying hypothesis of GWAS is common disease, common variants. So this is very, very important. Okay? And so GWAS generally do not capture rare variants. And there are many successful story about the GWAS findings. So you can see that this is in 2010, that is pretty early, that they summarized back in those days how many positive markers being identified related to different diseases. So the colors will represent different diseases, and then this clearly tells you where are the genetic locations are. But those are back in 2010. And this resource is actually currently being actively maintained. And if you go to this website, actually I just did a screenshot of this website 2 days ago. And so basically you will have much updated information, and you can select the disease you are interested in by typing in here, and you can set up your own filtering p-value threshold, and then to get to different stages. But bottom line is we have seen a lot of very important findings out of the GWAS study. The GWAS study comes with a lot of limitations. Okay, so the major limitation that people are talking about is lack of functional information. All right. So, which means disease trait associated SNPs are not necessarily causative variants. So what that means is if a genetic variant is associated to that disease, it's good as a marker, but that doesn't mean that SNP is causing that particular disease. So that is the general idea. So this is talking about this concept of haplotypes. And those are the SNP locations in the GWAS studies. They actually really doesn't tell you all the SNPs within these regions. But because these regions are so close to each other, this one will represent all the other ones. Okay, so that's why they call it something called a tag SNP. In these LD regions, with the causative variants serves as a genetic marker for this region, not necessarily representing every single one. So identification of the causative variants would require further studies, meaning that if I really want to find out the causal SNPs, I need to sequence very deeply for the entire region to find out every single SNP in this region. Only these markers probably doesn't work. And so there is a concept called something called missing heritability, which means when you identify the variants in the GWAS, typically only account for a proportion of heritability of the disease risk. So there is the one of the example is median odds ratio per copy of the risk allele is only 1.33, meaning that if I carry this SNP, I only have a little bit higher probability risk to get the disease than not. So there's a lot of reason was not considered in the GWAS studies. So there are many potential sources of missing heritability, including the SNPs with a prevalence of less than 5%, because if we do the GWAS, we only focus on the common SNPs. There are a lot of rare SNPs that are not being accounted for. So including the ones that it's very rare and it's relatively rare or very rare variants. And in the southern genome, and also many of them are not on the SNP arrays as well. We have never measured those. In the Southern Genomes Project, there are more than 11 million novel SNPs was identified. If we use the GWAS, they are not going to be identified at all. So the interactions between genes and another reason will be there is gene-gene interactions. It's not only one gene is causing the phenotype, but multiple genes. And there is a gene and environmental impact as well. And there's and also the contributions of structure variations that GWAS is not going to get us. You need to have the next generation sequencing to look into the structure variations. Okay, so let's take a 5 minutes break, and then we'll come back like 4:30.